In May 1952, in a basement laboratory at King's College London, something remarkable emerged from a photographic developing tray. A piece of film, a pattern of dark smears and arcs. The result of 62 hours of X-rays passing through a thread of wet fiber barely thicker than a human hair. To most people, it would look like nothing. Shadows on paper. But to a trained eye, to someone who spoke the language of X-ray crystallography, that pattern was almost unbearably clear. It showed a helix, a spiral. Not just any spiral, one with precise, measurable dimensions that encoded the structure of the molecule that makes you who you are. The scientist who made it knew exactly what she had. She was building, carefully, systematically, toward the full analysis that would let her publish it with complete confidence. She didn't know that within months, this photograph would leave her laboratory without her knowledge, and help win a Nobel Prize she would never receive. This is the story of Photograph 51, one of the most consequential images in the history of science. It's a story about one of the greatest discoveries ever made, and about the complicated, imperfect, deeply human way that discovery actually happened. The molecule at the center of it all was deoxyribonucleic acid, DNA, the instruction manual for all known life. Present in virtually every cell of your body, coiled so tightly that if you stretched out the DNA from a single human cell end to end, it would run roughly 2 meters, and yet it fits inside a nucleus you cannot see with the naked eye. By the early 1950s, scientists knew DNA was likely the carrier of hereditary information. They did not know its structure. Without its structure, they could not understand how it worked. This was the central unsolved problem in biology, and several teams in several countries were racing to crack it first. Watson and Crick in Cambridge, Rosalind Franklin and Maurice Wilkins at King's College London, Linus Pauling at Caltech in California. 3 groups, 1 problem, and a photograph that would tip the balance, not by being outright stolen, but through a chain of decisions and oversights that allowed it to travel beyond the woman who made it. The full story begins not in 1952, but 80 years earlier, in a medieval castle in Germany, in rooms that had once served as a kitchen, with a young scientist holding used surgical bandages from a nearby hospital and asking what, exactly, was inside the nucleus of a living cell. He would find something no one had ever isolated before. He would name it. He would not understand what it would mean, and the world would not understand it for another 70 years. Friedrich Miescher was 25 years old in 1869, working under his supervisor Felix Hope, a sailor, at Tübingen University. His question was specific. What is the nucleus of a cell actually made of? It sounds simple. At the time, cells were still poorly understood, and their nuclei were almost entirely mysterious. To get enough nuclear material of the study, Miescher needed cells with large prominent nuclei, and a lot of them. He found his source at a nearby clinic. White blood cells in wound dressings, pus cells, have large nuclei, and used surgical bandages were available in quantity. Not elegant, brilliantly practical. What he extracted from those cell nuclei surprised him. It was nothing like a protein. It was rich in phosphorus, an element not typically found in proteins at all. He called it nuclein. It was, we now know, DNA. The first human being ever to isolate it was a 25-year-old in a repurposed castle kitchen, working with material from surgical wound dressings. Hop. Saylor was cautious enough to hold up publication for 2 years while he repeated the experiments himself. Even then, neither man grasped what nuclein would eventually mean. The scientific world filed it away, interesting chemical curiosity, no obvious practical importance. The reason DNA was ignored for so long was logical. Proteins seemed far more likely to carry hereditary information. Proteins are enormously complex and varied. They are enzymes, structural materials, signals, doing almost everything in a cell. DNA, by contrast, seemed monotonous. Just 4 repeating bases. How could that encode anything? For roughly 70 years, this assumption held. DNA was the dull supporting player. And then, in 1944, at the Rockefeller Institute in New York, a quietly methodical bacteriologist named Oswald Avery set out to settle the question and upended everything. Avery, working with his colleagues Colin MacLeod and Macklin McCarty, studied a biological puzzle. A harmless strain of pneumonia bacteria could be permanently transformed into a dangerous one by mixing it with an extract from killed, dangerous bacteria. Something was being transferred, something that rewrote the cell's identity. But what? They destroyed each class of molecule one by one, proteins, lipids, carbohydrates, and tested whether the transforming effect survived. Each time, it did. Then they used an enzyme that specifically destroyed DNA. The transformation stopped. The conclusion was inescapable. The transforming principle was DNA. Genes, apparently, were made of the molecule everyone had written off. The paper was published in February 1944. The language was maddeningly cautious. This was revolutionary enough that Avery did not want to overreach. Not everyone believed it immediately. Skeptics worried about trace protein contamination. But the dam had cracked, and a biochemist in New York was about to drive a stake through the old assumption. Erwin Chargaff was an Austrian-born biochemist at Columbia University who read Avery's paper and had the kind of reaction that changes careers. If DNA was the genetic material, then understanding its chemistry precisely suddenly mattered enormously. He spent the late 1940s carefully measuring the relative amounts of the 4 DNA bases, adenine, thymine, guanine, and cytosine, across many different organisms. What he found, and published around 1950, was a pattern so clean it bordered on elegant. In the DNA of any organism, the amount of adenine always equaled the amount of thymine. The amount of guanine always equaled the amount of cytosine, no matter the species, no matter the tissue. A equals Tg equals C. Always. These became known as Chargaff's rules. Nobody quite knew at the time what they meant structurally. They were a numerical clue without a physical explanation, a locked door waiting for a key. That key would come from Cambridge, from a model built partly on data that wasn't the model builders to use. But we're getting ahead of ourselves. By the early 1950s, the race had genuinely heated up, and the favorite was not in England at all. On the west coast of America, at Caltech in Pasadena, Linus Pauling was the most celebrated structural chemist alive, and he had just pulled off a stunning coup. A year earlier, Pauling had worked out the structure of the protein alpha helix, not through exhaustive experiments, but through bold, intuitive model building. He knew the chemistry cold, trusted his instincts, built physical models, and found the answer. The scientific world was astonished, and now Pauling had DNA in his sights. In early 1953, Pauling published a proposed structure for DNA. It was wrong, significantly wrong. He proposed a triple helix with the sugar phosphate backbone at the center in a configuration that would be chemically unstable. Watson and Crick read it with something close to relief. The window was still open, but only barely. James Watson, an American biologist barely in his 20s, and Francis Crick, a British physicist who had not yet finished his PhD, shared a small office at Cambridge's Cavendish Laboratory. What they shared beyond that office was a conviction. The structure of DNA could be solved by reasoning and model building if they had the right experimental data. They were not going to generate that data themselves. They would assemble what others found, reason about the chemistry, build models, see what fit. It was Pauling's method, elegant when it works, entirely dependent on having access to the best measurements. And the best measurements were being made somewhere else entirely. At King's College London. By a scientist who had not agreed to share them. At King's, the situation was complicated. 2 scientists were working on DNA in the same building, largely in parallel, rarely collaborating. The dynamic between them would shape the entire story of how the double helix was discovered and how credit for it was distributed. Maurice Wilkins was a physicist who had been working on X-ray studies of DNA since the late 1940s. He had helped pioneer the field at King's and thought of himself as the natural leader of the DNA effort there. Rosalind Franklin arrived in January 1951, recruited specifically for her expertise in X-ray crystallography. From the beginning, there was a misunderstanding about what her role was meant to be. Franklin understood she had been appointed to lead an independent research program using X-ray crystallography on DNA. Wilkins, it seems, initially assumed she was a technical collaborator in his broader project. They were never able to fully resolve that confusion. They worked on the same molecule in the same building, largely in separate lines rather than together. What is beyond debate is the quality of Franklin's science. She was a world-class X-ray crystallographer who brought exceptional technical rigor to her work. Her camera setups were meticulous, her humidity controls, critical for getting clean, informative images of DNA, were precise and carefully maintained, and she made a key conceptual distinction that put her ahead of everyone else in the field. Franklin recognized that DNA existed in 2 distinct physical forms. A dry, compact form she called A form, a longer, more hydrated form, the one DNA adopts inside living cells, she called B form. Other researchers had noticed this difference, but Franklin was the most disciplined in separating them and studying each independently. Mixing them up produced blurry, misleading images. She established that B form was the biologically relevant one, the form DNA actually adopts in a living organism, and she set about studying B-form DNA with the systematic thoroughness that defined her approach. Control everything, measure everything, draw conclusions only when the evidence is solid. Others were more willing to guess. Franklin was not. Her PhD student Raymond Gosling had been working on DNA X-ray images since before Franklin arrived at King's, initially under Wilkins. When Franklin joined the lab, Gosling became her student and collaborator. Together, they were building the most precise X-ray portrait of DNA that had ever been attempted. Meanwhile, the competitive pressure was intensifying. Pauling's alpha helix triumph had shown what model building could achieve. Watson and Crick were building, discarding, and rebuilding models in Cambridge, waiting for data solid enough to build on. Chargaff's rules were out there, unexplained. And at King's, one of the clearest minds in structural chemistry was working toward the answer. alone. In early 1952, Watson had visited King's and seen some of Franklin's preliminary work. By his own later account, he didn't always take her seriously in the moment. That was an error of judgment that the historical record has consistently corrected. Franklin was not stuck. She was being careful. To understand why photograph 51 mattered so much, you need to understand, briefly, how X-ray crystallography works. It's the central technical tool of this story, and once you see the logic, the photograph becomes legible. So let's look at it. A sample, in this case a carefully prepared fiber of DNA, is mounted in an X-ray beam. X-rays are a form of light, but with a wavelength roughly the size of an atom, small enough to interact with the atomic structure of a molecule rather than simply bouncing off it. When X-rays hit the atoms in the molecule, they scatter in directions determined by how those atoms are arranged. Where atoms are regularly spaced, as in a crystalline or semi-crystalline structure, the scattered beams reinforce each other in certain directions, creating bright spots or arcs. Where they cancel, there's darkness. The resulting pattern is a direct fingerprint of the molecule's geometry. That fingerprint, the diffraction pattern captured on photographic film, must then be decoded, worked backward from the pattern to the shape that produced it. This requires deep expertise and careful calculation. It is not a photograph of a molecule. It is a mathematical shadow of one, in a language that takes years to learn to read. A helix, a spiral structure, produces a very specific signature in diffraction, an X-shaped cross. The angle of the cross arms encodes the angle of winding. The spacing of the dark horizontal arcs at top and bottom encodes the distance between repeating units. Each feature of the pattern is a measurement, if you know how to read it. Photo 51 showed that cross sharply, cleanly, with a clarity nobody had captured before. The B form of DNA, at very high humidity, around 92%, produced an almost ideal diffraction pattern. The X-shaped cross was unambiguous. The arc spacing encoded precise helix dimensions. Franklin, when she examined it, knew immediately it was exceptional. From that pattern, Franklin could measure, and did calculate, the key structural parameters of DNA in its B form. The diameter of the helix, the distance the helix rises per base pair, the number of base pairs per complete turn, the position of the backbone. Each number was a constraint that any valid model had to satisfy. She was not ready to publish. She wanted to be certain. Her notebooks show the progression, calculations, recalculations, cross-checks. She knew she had the best X-ray image of B-form DNA ever taken. She was building an airtight case. She was weeks or months from a complete, defensible, published account. She intended to do this on her own terms. In December 1952, a Medical Research Council committee visited King's College for a progress review, a routine annual event for publicly funded labs. The committee received a written report summarizing the work underway at King's, including Franklin's crystallographic measurements and key calculated parameters. It was an internal document, produced for funding oversight. It was not a published paper. It was not data Franklin had offered to share with competing researchers. It was an administrative summary. One of the committee members was a Cambridge crystallographer named Max Perutz. He took the report back with him. In early 1953, Perutz shared the MRC report with Watson and Crick. The report contained Franklin's detailed measurements of the DNA unit cell, including the precise crystallographic symmetry of B-form DNA. Crick, who understood crystallographic symmetry deeply, recognized immediately what it meant. The two strands of the helix must run in opposite directions. Anti-parallel. That was a structural constraint that went directly into their model. Perutz later acknowledged he had not thought carefully about whether sharing a report was appropriate. There was no specific rule that forbade it. But Franklin had not been asked. She did not know her unpublished measurements had traveled to Cambridge. She had not agreed to share them with competing researchers working on the same problem. A separate breach occurred during Watson's January 1953 visit to King's College. He had come to see Wilkins. Franklin was not part of the conversation. Wilkins, according to Watson's own account in The Double Helix, published in 1968, retrieved a photographic print of Photo 51 and showed it to Watson without Franklin's knowledge or consent. Watson later described what he saw, the X-shaped diffraction cross that could only come from a helix. The layer lines that encoded the helix dimensions, the numbers that told him how wide the molecule was, how tightly it coiled, how many strands it contained. He memorized the measurements. He returned to Cambridge. He told Crick what he had seen. Were these acts of deliberate theft? Historians have examined this carefully and at length. What seems clearest is that no single person set out to steal Franklin's data. There was no conspiracy. There was instead a culture, one in which data produced in one lab routinely flowed to friendly colleagues in another, without always considering whether the person who generated it had been asked. That culture affected Franklin because she was, in some ways, outside it. She was not part of the informal network of conversations at conferences and lunches where data circulated freely. She was working in a relationship with Wilkins that was tense and non-collaborative. Her data was excellent, and it was accessible to people who had not made it. The ethical failure was real, regardless of intent. Photo 51 and the MRC report measurements were both material to the model Watson and Crick built. Their Nature paper acknowledged stimulating discussions with the King's College group. The actual flow of information was more consequential than that phrase suggested, as Watson himself would acknowledge in print 15 years later. Franklin did not learn during her lifetime the full extent to which Photo 51 had been shown to Watson. The complete picture of what had happened became clearer only with Watson's 1968 memoir and through subsequent historical research. By the time the book appeared, Rosalind Franklin had been dead for 10 years. Back in Cambridge in early 1953, Watson and Crick had the data they needed. Photo 51's dimensions, Franklin's unit cell measurements from the MRC report. The double helix was nearly within reach. But there was one remaining obstacle, and it came from cardboard cutouts on a desk. Watson was building physical cutout models of the 4 DNA bases: adenine, thymine, guanine, cytosine. He was using shapes drawn from standard chemistry textbooks. And those shapes were wrong. The textbooks showed the bases in what chemists call the enol form, one possible configuration of atoms around the molecule's hydrogen bonding positions. The correct form was different. The person who set him straight was sitting a few meters away. Jerry Donahue was an American crystallographer sharing their Cambridge office, a scientist with deep hands-on experience with organic crystal structures and a former member of Linus Pauling's Caltech group. He had been watching Watson's cardboard puzzle with growing impatience. Donahue told Watson plainly, "Those tautomeric forms are wrong." The organic chemistry literature was littered with unreliable textbook diagrams. The DNA bases, in real molecules, exist predominantly in the keto configuration, not the enol. He had seen it in crystal structures. Watson was initially skeptical. Then he cut new cardboard shapes and tried it. The keto form shapes clicked together in a way the enol shapes never had. Adenine locked onto thymine, held by 2 hydrogen bonds. Guanine locked onto cytosine, held by 3. And both pairs were almost exactly the same width, meaning the double helix could be uniform in diameter along its entire length. The geometry was perfect. And something else clicked simultaneously. This base pairing explained Chargaff's rules. If every adenine must pair with a thymine and every guanine with a cytosine, then the amounts of A and T in any stretch of DNA must always be equal. Same for G and C. The numerical pattern Chargaff had measured without knowing why. The structure answered it. Watson and Crick worked at a feverish pace over the following days, building their metal plate model. 2 strands, anti-parallel, running in opposite directions, a feature they owed in part to Franklin's MRC report data. The sugar phosphate backbone on the outside, as Franklin's X-ray images had shown, the paired bases facing inward like the rungs of a twisted ladder. The breakthrough date Watson later identified was February 28, 1953. That afternoon, according to his account in the double helix, Crick walked into the Eagle, the Cambridge pub where Cavendish scientists regularly took lunch, around the corner from the lab, and announced to the room that he and Watson had discovered the secret of life. Watson's account doesn't claim the words were verbatim, but the moment is commemorated on a plaque at the Eagle today. Whether those exact words were spoken exactly that way or not, something real had happened. They had a model for DNA, a double helix, that explained the X-ray data, satisfied the chemical rules, matched Chargaff's ratios, and revealed, in its very shape, how hereditary information could be copied exactly every time a cell divides. The mechanism of life was in the structure. Watson and Crick wrote up their proposed structure in a letter to the journal Nature, barely 900 words, one of the most concise announcements in the history of science. Near the end, they noted that the base pairing arrangement had not escaped their notice. It immediately suggests a possible copying mechanism for the genetic material. One sentence. The logic of heredity explained. On April 25, 1953, Nature published 3 papers back-to-back in the same issue: the Watson-Crick double helix proposal, X-ray data from Wilkins, Stokes, and Wilson, and crystallographic data from Franklin and Gosling. The 3 together told the story: here is the proposed structure, and here is the experimental evidence it rests on. The arrangement subtly obscured something. Franklin and Gosling's paper, with rigorous crystallographic data that had, in fact, been a foundation for the model, was positioned as independent confirmation, appearing as though it came after Watson and Crick's model rather than before it. The actual chronological flow was more complicated. Franklin and Gosling's paper was solid, correct, and important science. Years later, Franklin herself, having moved to Birkbeck College to work on tobacco mosaic virus, acknowledged the double helix as the correct structure and worked productively alongside Watson and Crick in the broader scientific community. The professional rift at King's had faded. The damage to her credit had not. The scientific community received the triple papers with growing appreciation over the following months and years. The double helix was elegant. It explained the data. Chargaff's rules fell out of it naturally. Further experiments confirmed it. By the late 1950s, it was established beyond serious doubt as the correct structure of DNA, and one of the great intellectual achievements of the century. What the structure meant in practice, each strand of the double helix is an exact template for rebuilding the other. When a cell divides, the helix unzips, and each strand serves as a guide, complementary bases arriving and bonding, a pairing with T, G pairing with C, assembling two identical double helices from one. Replication is written into the shape of the molecule itself. Rosalind Franklin left King's College in 1953. She had already arranged the move before the triple papers appeared in April. She went to Birkbeck College, London, where she turned her skills to the structure of tobacco mosaic virus. The work she did there, on the structure of plant viruses, was outstanding science on its own terms, recognized by her colleagues as first-rate. She did not, as far as we know, learn during her lifetime the full extent to which Photo 51 had been shown to Watson without her consent. The picture of what happened became clearer only with Watson's 1968 memoir, The Double Helix, which described the episode with Watson's characteristic candor and his equally characteristic lack of diplomatic concern for how it would reflect on his treatment of Franklin. Watson's portrayal of Franklin in that book was, to put it plainly, damaging and inaccurate. He referred to her by the nickname Rosie, a name she never used. He depicted her as unable to interpret her own X-ray data, as an obstacle rather than a contributor. His account shaped public perception of Franklin's role for years. Historians have spent decades correcting it. What her laboratory notebooks, her publications, and the testimony of colleagues who worked alongside her actually show is something quite different, a scientist of exceptional ability doing exactly what a scientist of her caliber should do, building a meticulous, defensible case before putting it in print. She had not failed to understand Photo 51. She understood it precisely. She was waiting until she could prove it. In 1956, Rosalind Franklin was diagnosed with ovarian cancer. She was 36 years old. She underwent 3 operations and experimental chemotherapy. Through most of this, she continued to work, publishing papers, attending international conferences, completing significant research on biostructure. Her colleagues at Birkbeck later described her as extraordinarily committed, even through serious illness. On April 16, 1958, Rosalind Franklin died of ovarian cancer. She was 37 years old. 4 years later, in October 1962, the Nobel Prize in Physiology or Medicine was awarded jointly to Francis Crick, James Watson, and Maurice Wilkins for their discoveries concerning the molecular structure of nucleic acids and its significance for information transfer in living material. 3 names on the certificate. The Nobel Prize has not been awarded posthumously since a rule change in 1974, and even before that, posthumous awards were extremely rare. Franklin was simply ineligible. She had not been passed over. She could not have been included. The injustice, to the extent there was one, lay in the events of 1952 and 1953, not the Nobel Committee's decision in 1962. The ethical questions around the double helix story are legitimate and important, and they're best asked precisely not why didn't Franklin win the Nobel? She was dead before it was awarded, but was her data used without her knowledge to advance a discovery she did not share in? The answer to that is yes. That is established by Watson's own account. A 2023 article in Nature by historian Matthew Cobb re-examined the historical record and emphasized that Franklin was a major figure in the DNA story, not a passive victim, but an active scientist whose work was essential to the discovery. Both things are true at once. She was formidably capable, and her work was used without her knowledge. Holding both truths without simplifying either is how we understand history honestly. Watson and Crick made a genuine, brilliant discovery. Franklin's work was essential to that discovery and was accessed without her consent. These facts do not cancel each other out. They are both part of the story. What we can say with confidence is this: without Franklin's photograph, without her measurements of the unit cell, without Donohue's correction, without Chargaff's rules, without Avery's transforming principle, without Miescher's nucleon, the double helix would not have been built when it was, and possibly not by Watson and Crick at all. Science is a relay race. She ran her leg faster than anyone. Rosalind Franklin's name has been given its proper weight in the decades since. King's College London opened the Franklin Wilkins Building in honor of both scientists. The Royal Society established the Rosalind Franklin Award in 2003, given annually for outstanding contributions to natural science or engineering. The Rosalind Franklin Institute, a national research center for life sciences, opened in 2018. And in 2019, the European Space Agency named its ExoMars rover after her. The Rosalind Franklin rover is designed to drill up to 2 meters below the Martian surface, searching for biosignatures of past life. The scientist who helped reveal life's molecular blueprint now searches for life on another world. As for what the double helix itself unlocked, the list is almost too large to catalog. Once you know the structure of DNA, you can begin to read it. The genetic code, the rules by which sequences of 3 DNA bases specify particular amino acids, which build all proteins, was cracked within a decade of the 1953 papers. By the 1970s, scientists could cut DNA at specific sequences and splice in new pieces, the foundations of genetic engineering and biotechnology. By 1990, the Human Genome Project had begun the task of reading all 3 billion base pairs of the human genetic blueprint. It was completed in 2003, 50 years, almost to the month, after the double helix papers. Sequencing DNA today is so fast and cheap it is used routinely in medicine, to identify cancer mutations, to trace the spread of infectious diseases, to diagnose rare genetic conditions in newborns, to match donors and recipients. The double helix is not a historical artifact, it is operational infrastructure for modern medicine. And then there is CRISPR, the gene editing system that allows scientists to find a specific sequence in a living cell's genome and edit it with precision that would have seemed like fantasy in 1953. CRISPR is used in medicine, in agriculture, and basic research. It is only possible because we understand exactly what the double helix is, how it's structured, and how it works. All of this, the sequenced genomes, the engineered bacteria that produce insulin, the forensic tests, the pandemic response vaccines designed from genetic sequence data in days, traces back through time to a February morning in Cambridge when cardboard shapes clicked together on a desk, and further back still, to 62 hours of x-rays in a basement on the Strand, and further back still, to Irwin Chargaff counting bases at Columbia, to Oswald Avery, Colin MacLeod, and Macklin McCarty coaxing bacteria to give up their secrets, to Friedrich Miescher in a castle kitchen in Tübingen, puzzling over something phosphorus-rich found in white blood cells from a surgical ward, and calling it simply nuclein. Science builds this way, on unremarked work, on preliminary findings that don't make the headlines, on careful measurements taken by people who never received the prize. The Nobel-winning discovery at the end is the visible tip of a very long story, one that includes everyone who contributed a fact, a number, a photograph, a correction. Even a correction about the tautomeric form of a base from a crystallographer at a nearby desk. The story of Photo 51 is not a simple story of heroism or villainy. It is a story of human beings doing their best and their worst, sometimes simultaneously, operating in institutions and cultures that had not caught up to fairness, driven by ambition and genuine curiosity in roughly equal measure, and producing, despite all of it, one of the greatest discoveries in the history of science. Rosalind Franklin was not a martyr. She was a scientist, a formidably good one, who did her work with precision and integrity, and whose contribution to one of the century's defining discoveries was greater than she was given credit for during her lifetime. Her reputation is now in a place much closer to where it belongs. It took decades. The record has been corrected. The universe keeps its best secrets in the shape of things, in the angle of a crystal, in the spacing of arcs on a piece of photographic paper, in the way 2 strands wind around each other in opposite directions, held together by pairs of bases that fit like perfect complements. Each A matched to its T, each G matched to its C. That shape, wound, paired, anti-parallel, is the shape of life. It is in every cell in your body, right now. Your DNA is replicating this moment, somewhere in some dividing cell, because the structure of the helix makes replication possible. 1 strand as the template, complementary bases arriving and bonding, the process so reliable it runs billions of times every second across the trillions of cells in a human body. Miescher found nuclein in 1869 and didn't know what it was. Avery showed it carries heredity in 1944 and wasn't fully believed. Chargaff found the pattern in 1950 and didn't yet know what it meant. Franklin captured its clearest image in 1952 and was still working toward its full interpretation. Watson and Crick assembled the pieces in 1953, and the world changed. That is how science works, not in isolated flashes of lone brilliance, but in accumulation, in conversation and competition, in correspondence and mistake, in data passed across tables and photographs seen through file cabinet glass, in all its messy, imperfect, unstoppably productive human glory. And somewhere in that long chain of people asking what things are made of, how they are shaped, what the shape means, 1 photograph. 62 hours under an X-ray beam, a cross of dark arcs on white paper, photograph. 51. The image that helped reveal the molecule that makes you you. And in that molecule, in those anti-parallel strands and paired bases, is something that makes this story feel right rather than merely bittersweet, the logic of recognition. Every A recognizes its T. Every G recognizes its C. In the double helix, complementary things find each other. They always do. Even if sometimes it takes a little longer than it should. thanks for spending this time being curious with me. If the story of Photo 51 left your mind in a slightly different place, if you're thinking about credit and discovery and what science is actually like underneath the textbook, good. That's exactly what it's supposed to do. Until next time.